I'm so excited. Chapter 33 and 34 are my favorite chapters. Okay, so chapter 33 is broken up into two pieces because it's a huge chapter. And what we're going to be doing at this point is getting into all of the different phyla and classes that are out there. And there are a lot, I'm not going to lie to you. So what we're going to do is start with the least advanced organisms and then move up to the most advanced organisms. And that's kind of how we're going to go. So what we're going to start out with are what are called non-coelomate invertebrates. So let's think about what that means. Non-coelomate is acoelomates, right? So they don't have a body cavity, and they are going to be invertebrates, meaning they don't have a spinal column. Well, yeah. Okay, so let's talk about sponges. That's going to be the first phylum that we're going to look at. So sponges are going to be phylum Porifera, and the name pori means with holes, and that makes sense because sponges have holes. And so this is going to be a pretty big phylum, over 5,000 species. Most of them are going to be in marine environments. There's a few that are freshwater. These ones are going to be sessile, and what I mean by sessile is that they are going to be stuck to the ground, right, or the substrate that they're growing in. So if we take a look at this right here, this is a picture of a sponge right here, and not a lot of symmetry, right? It's not exactly symmetrical, and that's why it's in the group Parazoa instead of eumetazoans, right? Sponges can get to be very big. So here are some sponges right here. These are sponges right here. This is sponges right here. These are all coral, but anyway, sponges can get to be very huge. There's a type of sponge called a barrel sponge that I've been completely inside of because, long story, I was hiding from a shark. But anyway, so they can get to be very big. They can be very small, but they're very sensitive to the environment. And if you expose them to air, a lot of sponges will actually die after that. They'll go septic, so it's not a good thing. Now let's look at their actual shapes. One thing that you may notice with these and these here and in this previous one is that they all kind of have a vase-like shape with that big hole at the top. So that hole at the top is called the osculum, and that's going to be where particles are going to flow out of. They're going to pull water in through the sides, and then it's going to come out through the top. So same here, water will go in through the sides and then out through the top here. So let's look at a diagram of the way that they look. So here's our osculum, that big hole at the top. And remember I talked about those choanocytes. Look at that, how similar that looks. So that's why some scientists say that maybe this was a bunch of these guys that ended up living together. They used to be protists and now they're going to be these choanocytes inside of a sponge. Who knows? So the sponge is going to be very basic. It's going to have three layers. So you've got this innermost layer, which is going to be made of the choanocytes. And then you can see these blue and yellow things around here. And those are going to be, the blue ones are going to be amoebocytes. And amoebocytes are going to actually look like amoeba and kind of move around like amoeba. And they're going to secrete spicules, which are going to be the yellow that you see here. And spicules, if you look on this next phrase, photo right here, spicules are going to be made of silica, and they're going to have a couple of different purposes. One of them is they're going to prevent predators from coming in and biting them, right? And then another one is they're going to be structural components. They're going to help them to be able to grow tall by providing some sort of structural integrity to the sponge. Now, there are some sponges called glass sponges, like you see here, and those are tons and tons of spicules, and then there's some that don't have a lot. But glass sponges are just really pretty and cool to look at. So those are going to be the parts of the sponge. The last thing is that if you look in this picture here, nope, here, they have an outer layer, which is just epithelial cells. They're just kind of flattened cells that are a little tougher on the outside of the organism. Okay, now let's go to talking about how these guys reproduce. They can reproduce sexually or asexually. In this picture, you can see that they're reproducing sexually. And so what that means is they're going to produce gametes, male and female gametes, throw them into the water, and hope that they find the opposite sex. That's it. So you can see that's exactly what this one is doing. It's throwing all its gametes out of the osculum. Now, another thing that can happen is they can actually fragment, which is where you're going to have a little piece break off, and then that'll turn into a whole new sponge itself. I've had experience with both of these situations, actually. I was scuba diving to do one of my fish counts that was for my research, and one of these sponges just, I don't know, conditions were right, and it just started throwing up all of these gametes, but they were huge, like those big fluffy snowflakes that we get in the spring here. And they just started going out of control, and I couldn't even see more than my hand in front of my face, and I was like, all right, that's it, I'm out of here. And then I also had another experience where I was trying to do a fish count, and the current was really strong. It was way too strong for us to be out there, but I wanted to finish my research. So we went out there, and I got to the bottom, and I couldn't even stand up because the current was so strong. I mean, my face was jiggling and everything. And so I found a sponge that actually looks like the one in this picture here, and I kind of wedged my butt into it to kind of stabilize myself, not thinking about the huge scuba tank on the back. And so what happened was that broke a chunk off that fell down, and I ended up, finishing my dive, and I ended up having to come back and redo that dive because it was an outlier because the conditions were so different from all the other times we did counts. And when I went back down there, there was that little baby piece of a sponge, a fragment just starting to grow. So I helped it asexually reproduce is the way that I like to think about it. Okay, so those are going to be sponges and the way that they reproduce. So I'm going to move down here. All right, so we're already at the radially symmetrical organisms. So if you remember, radially symmetrical means they're usually going to be circular. They have that central point, and you can draw a line through that central point anywhere, and you'll have a mirror image. That's different from us with bilateral symmetry. So there's going to be two phyla that are going to be in this group. Phylum Cnidaria and phylum Ctenophora. Notice the C is silent in both of those. Okay, phylum Cnidaria. Cnido literally means stinging. So these are going to be organisms that can sting. Ctenophora we'll talk about in a little bit. So most of the cnidarians are going to be marine, and they're just basically gelatinous. This is going to be jellies, sea anemones, corals, those types of things. They are carnivorous. Most of them do tend to be sessile. If you think about corals or anemones, they're stuck to the bottom. They get their prey with their tentacles, and then the tentacles are usually in a ring that's surrounding their mouth. They're going to have free-swimming larvae that are called planula, and they're usually going to be released into a current because even though they have cilia, these guys are microscopic. They're not going to get very far unless if they're in a current. They are going to eat through what's called internal extracellular digestion. And what that means is it is internal, so it's inside of them, and extracellular means that they're going to secrete enzymes out of the cells and just digest whatever it is. They don't have a brain. They don't have any sort of respiratory system, circulatory system. There are some studies that say they have a little bit of nervous tissue, but not really anything to speak of. Okay, so the reason these guys are called cnidocytes is because they're going to have stinging cells on them called nematocysts, and I'll show you a picture of that in just a second. But before we do that, let's take a look at some organisms that are in this group. Okay, so, I do have some pictures we'll look at in a little bit. Okay, so here we have the cnidocyte, and cnido means stinging, cyte means cell. Within the cnidocyte is going to be something called a nematocyst, and this is what's actually going to cause the sting. So these are going to line the outside of all the tentacles of these guys, and they're going to have a little trigger that's sitting on the outside of them. What's going to happen is if you touch that trigger, that's going to cause them to pump water into the nematocyst, and that's going to throw this thread out, and this thread has got little hook-like barbs on it, and they're going to stick into whatever caused it to trigger, and that's going to develop, or that's going to distribute poison into whatever it was that set off the trigger. Now, some people can touch an anemone, no problem. They can touch coral, no problem. And that totally depends on whether you're allergic or not. I am severely allergic to these guys. I can't remember if I downloaded the pictures on this PowerPoint, but I'm extremely allergic to these guys, and I've got some gnarly pictures that I'll show you. So anyway, that's going to be the cnidocyte that's going to be really, 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 really important. Going on to this next slide is going to show you, the different life stages that these guys can have. So they can have this, which is going to be the polyp stage, or they can have this, is just called the medusa stage. So the polyp looks more like a plant where it's stuck to the bottom and the tentacles are sticking up. And the medusa has that bell shape with the tentacles hanging down, very typical of like a jelly. So those are going to be the two forms that they can have. Now, depending on the species, the class, all of that stuff, they can start out as a polyp and then end up as a medusa. They could start as a medusa and end up as a polyp. They can only be medusa, only be polyp. So there's a lot of variation as far as that goes. So in this next one, we're going to get into some more of the classes of these different guys.